Looks great. Yep. Thank you.
Thank you. Yeah, great. So it's really nice to be with you today to share some of the work my group has been doing on microbial ecology in Lake Champlain broadly. The story I have tried to spin for you today is about sort of these seasonal dynamics that we're looking at. And with a slight caveat that these data are a bit hot off the press. So we'll see what we see today, and certainly some ongoing sort of questions that form as we go along. And this work has been conducted at Middlebury College with my research students. We're an undergraduate only institution. So all of the work I'm presenting today has been conducted by my team tiny crew over some field seasons. And I just want to start by making the Middlebury College land acknowledgement. So Middlebury College sits on land which has served as a site of meeting and exchange among Indigenous peoples since time immemorial. The Western Abenaki are the traditional caretakers of these Vermont lands and waters, which they call Indokina, or homeland. And we remember their connection to this region and the hardships they continue to endure, and we give thanks for the opportunity to share in the bounty of this place and to protect it. And I start with that acknowledgement to really emphasize that the work we've conducted is in these ancestral homelands, both the water and the land that we've conducted this work with. So back to that title a bit to unpack and give a bit of background. So the first part of this title is engaging with these idea of seasonal dynamics in Lake Champlain, and of particular interest in many temperate lake ecosystems are changing winter conditions. And so these images we'll work through are from some really interesting sort of assembly of information from the lake ice continuum concept put together by Cavalier and colleagues in this paper. And winter changes are particularly of interest because we think, although there are other changes we're noticing, for example, summers of drought or extreme flooding, many of the changes in these systems are coming in winter in terms of changes in snow and ice cover on parts of lakes. And so these dynamics can shift in terms of light availability when there's snow on top of ice, just ice, or open water. So as we see decreases in ice cover, we see increasing light availability, which can certainly impact the productivity in these ecosystems, and then sort of the nutrient dynamics there as well. And then also sort of important are these exchanges between the air, the atmosphere, and inputs, these exogenous inputs from terrestrial systems. And the changes in winter are really interesting in this way because they're shifting from the sort of historical or perhaps current lake seasonality where there's low land and atmosphere connectivity to the lake, as well as fairly low light in the winter. And we're seeing this sort of shift to more connectivity and more light availability in the winter. So those are some of the sort of big picture changes we're interested in looking at winter in particular. The other piece of this title that may be less familiar to some is this idea of microbial ecology. So microbial ecology is like many disciplines within ecology. We're thinking about sort of who is there, how many of these different organisms are present, how are they interacting with one another, and what functions are they carrying out in the ecosystem. So this little model here is just sort of depicting that we might have several different taxa, and they interact with each other in different ways. Some of them are going to negatively impact neighbors. Some of them might have metabolites that are exchanged. And all of this is really interesting to sort of understand and unpack, but unlike perhaps studying fish or birds, where not that those projects are easy, but where we have a bit more visibility in terms of we can look at these communities and assess their different species and the functions they're playing out in these ecosystems. For microbes, we're a bit we have to use different tools to look at and answer some of these questions. And so the tools I predominantly use are molecular methods. So from a whole community of microbes, we can extract information, genetic information like DNA and RNA, and then we can sequence those biomolecules to then understand information about their identity. And you can do this in other larger organisms as well, but this is sort of a really key feature when most morphologies of microbes are short sticks or small circles, rods and cocci, and those morphologies are really poor predictors of their function in the ecosystem. And then for a little bit of context on the sort of subtitle of my talk here, this exploring the seasonal variation and linkages to algal blooms, cyanobacteria are a really important group of organisms in terrestrial and aquatic ecosystems in general. And we think about their contributions to the carbon cycle through photosynthesis. They pioneered this function, this ability to take in inorganic CO2, producing oxygen along the way and making this organic carbon, which is contributing to something like 25% of Earth's productivity still today. And it was a very evolutionary advantageous process that was taken up by early plants and has persisted through in chloroplasts today. This feature is particularly of interest. This oxygen production is what got us to the point of oxygen in the atmosphere that we have today. So they're deeply involved in carbon cycling. There also are a number of different cyanobacterial taxa that are involved in nitrogen fixation. So making this inorganic nitrogen in our atmosphere biologically available. So they take in inorganic nitrogen and fix it. They're not the only organisms that do this, but biological nitrogen fixation across terrestrial, agricultural, and aquatic ecosystems leads to inputs of fixed nitrogen at about 258 teragrams of nitrogen per year. It's a really important process for making nitrogen available in ecosystems. So cyanobacteria are really central to both carbon and nitrogen cycling, but in the context of cyanobacterial blooms, they become the sort of nuisance dominant taxa in these ecosystems and really severely impact ecosystem function. The harmful piece is typically due to some of the toxins they can produce. And there are a number of different conditions that can lead to these algal bloom features. So this is a photo from up in the Northeast Arm a few years back now, where there's just a full sort of slimy green cover on the top of the water. And there are a number of different conditions that can lead to these blooms. So this framing of some like it hot has been helpful for a number of years, thinking about warm water conditions, relatively calm low winds, so there's not a lot of mixing in the ecosystem, and then these inputs, these nutrient inputs from terrestrial systems, either sort of directly into the lake or via streams, creeks, rivers. More recently, there's a framing from Reinl and colleagues, two different papers actually. This paper is the one particularly thinking about some like it cold. There's another article looking at also blooms happening under low nutrient conditions. And so over this range of conditions, both in warm and cool climates, we increasingly are seeing cyanobacterial algal blooms. And so that's sort of the big framework of where our study is fitting in. So the objectives for these projects have been to characterize these microbial communities in Lake Champlain. For one of the water center grants we had, it was to look at samples before, during, and after bloom events to try to look at what's changing in these communities before, during, and after. And then the more recent project is to think about sort of over the course of winter, how are those communities changing? And then the sort of second part of this the study objectives have been to identify parameters within our data set. So the physicochemical features that we're measuring, as well as utilizing really lovely publicly available data sets to try to help estimate trends. So sort of model some of these trends that would help explain timing of these bloom events in different basins around the lake. And so in this first objective, part of the original task is just trying to identify, which bacteria, cyanobacteria, and viruses are present in this ecosystem. And so this will be the focus of what I talk about with you all today. And as a bit of a extension to this background, perhaps viruses aren't the top thing you think of when you think of aquatic ecosystems. And my training, my PhD training was largely in viral ecology, and so I'm a bit biased in this thinking, but viruses are really key players in a lot of freshwater systems and truly across ecosystems in general. So in this very simplified freshwater food web, we would have phytoplankton members that are bringing primary productivity into the system, and then also these heterotrophic bacteria and archaea, zooplankton and protozoan grazers, and then sort of up to these other levels, higher trophic levels. And if we think about where viruses fit and impact this food web, the spoiler alert is that they impact all of these different organisms. So the majority of viruses in freshwater ecosystems are infecting prokaryotic organisms, so bacteria and archaea, but there are certainly viruses of all of these types of groups of organisms, and they're really interesting to unpack and understand in these ecosystems because they can affect the pools of dissolved organic matter, and when they infect a host and that host lyses, there's also alteration in the availability and sort of layability of nutrients within these environments. So activity of these viruses can be another sort of interesting part of the microbial ecology to unpack to understand these systems. Okay, so our study has been focusing in some different locations around Lake Champlain. So here's sort of a generic map of the lake with different basins parsed out. And we've been, initially we focused at one site in the northeast arm to sort of pioneer some of these methods. And then subsequently we've been looking across seasons at a site in Missisquoi, in St. Albans Bay, and in Mallett's Bay, which capture that range of trophic status across these basins, which is also part of our curiosity, is understanding, how do these microbial communities vary based in these systems, and again, across temporal periods of time as well. So when we go out and sample, part of what we're doing is trying to characterize relative abundance of these organisms, and we do that through microscopy. And I could sit at a microscope all day looking at some of these cyanobacterial samples, but we use phase contrast microscopy to sort of identify, at least to genus, which cyanobacteria are present under bloom conditions. And then we also use epifluorescent microscopy, so relying on fluorescence signal to detect microbes in two different sort of flavors, where in one set of samples we add a stain, which is able to bind or interact with DNA, and that fluoresces really brightly. And so we can look at these little arrows are a bit hard to see, but these still small, but relatively, I guess they're the medium-sized thing that we're looking at in this image, are the bacteria. So we count those. These little pinpricks of light in the background are actually the viruses. So we're able to stain and count the viruses. We call them virus-like particles, because it's also possible that little bits of DNA are also just fluorescing here, but we get an estimate of virus-like particles. And then we rely on the inherent photopigments present in cyanobacterial to conduct counts of them as well. So we are able to provide an excitation wavelength and look at the emission from certain photopigments to count those. Beyond just counting them, we also take samples. So these are some of the team out in the field, Irene on Missisquoi when it's frozen, Bree and Gifty in a summertime sampling, and then this was just a few weeks back, Angie and Tanaya out. So we collect water in the field, we bring it back to the lab and filter on different pore sizes to capture different fractions that include the bacterial fraction, and then whatever's left behind, we concentrate and filter again to capture the virus-like particles. And then from there, we extract our molecule of interest, so DNA or RNA for the to look at activity of what's going on. We sequence the data, we get the data back and check for quality and then assemble it. And if anybody has sort of particular methods questions there, I'm happy to talk through a little bit more of that. But what I'd like to shift gears to now is what we've been seeing. So in the summer 22 data, so these are the data we collected where we looked at pre, during, and post bloom communities just at this one site. We have, in this data set, what we're looking at is the Bray-Curtis dissimilarity. So this is a measure of taxa abundance and their relative abundance and the total number of taxa within a given sample. And this type of analysis is able to reduce from this multidimensional space of trying to understand the relationships of these organisms into just two axes. And so what's being represented here, each of these data points represents the total diversity in one sampling time point in this location. The dates are a bit hard to see. I apologize for that. These data are actually currently on study abroad in Sweden with my student Irene. So this is just a screenshot she sent me. But the sort of interesting features we can pull out from this analysis are the ellipses that are representing the sort of confidence in clusters of these data. So in total diversity, what's interesting is that the pre-bloom samples are quite sort of diverse, and the bloom samples, we just have two in this data set, but they cluster a little more tightly, and this transition point seems to happen that after the bloom in these green sampling points, the community has sort of shifted from this pre-bloom data set. And so if we dial in a little bit deeper onto some of these data, there's a lot going on here, so we'll walk through it. We can unpack a little bit more about the taxonomy, so the different sort of species composition, as well as the function or predicted function of what's going on in those communities. And so these are heat maps. The warmer the color, the more abundant that taxa or functional gene is in a system. The cooler colors mean they're less abundant. The neutral 0 is sort of this light yellow. They're neither more abundant or less abundant between samples. What's being grouped or sort of represented at the top of each of these figures is the group. So in this case, this blue bar is indicating metagenome data. So that's the whole bacterial community DNA. The pinkish bar, salmon colored bar at the top here is representing the viral data. So the full composition of viral genomes that we were able to measure. And along the bottom are different sampling time points, but they roughly go from the beginning of the summer to the end of the summer, with our blooms occurring in the sort of later range. Okay, so what can we unpack from this? What are the differences that sort of start to stand out? I was very excited to see this line here for T4 virus. This is actually indicating these Myoviridae family, which are a really common, one of 3 common families of cyanophage, so viruses of cyanobacteria. So we're excited to sort of dig into some of those taxa a bit more in depth. This is at the phylum level of annotation, so it's really sort of a broad course metric. The really sort of fun things that I'm starting to look into are these differences here in this portion of the heat map are really indicating the variation in taxa at the phyla level over time in this one site. And we see, you know, some sampling points, there's, you know, a certain group that seems to stand out. But if I look and compare between taxonomy and these functional groups here, there are less drastic differences in abundance of those functional groupings than there are across taxonomy. So it seems like while the exact community members may vary, they seem to also sort of have these largely similar sort of functional capabilities, which is interesting if we're thinking about how these communities might relate to sort of nutrient cycling and processing in these systems sort of before, during, and after blooms. And then this top piece here is stands out in the functional piece as well. There are a number of highly enriched genes associated with carbon metabolism. So these codes here are indicating different metabolic functional groupings. And so I've just sort of highlighted these a bit here to show that the glycosyltransferases, the glycoside hydrolases, and these carbohydrate binding module functional genes seem to be enriched in our viral data set, which is also, in thinking about sort of carbon moving in these systems, is sort of intriguing. So we'll be looking into that a bit more as well. Changing gears a little bit here, looking at the counts data. So this now is from December, last December through July of 2023. Now looking at counts data across Missisquoi Bay, St. Albans Bay, and Mallett's Bay. So eutrophic, meso to eutrophic, and then meso to oligotrophic status. And the purple bars are representing our viral counts per mil. The orange bars are bacterial counts, and the green bars, which are relatively lower in abundance here at the base, are, say, cyanobacterial counts. And so these are just sort of informative. We can sort of unpack these a little bit more as we get our physicochemical data on board to look at what might be leading to shifts. But what sort of stands out to me in the sort of viral piece is that typically we think about viruses, there being about 10 viruses to every bacterium in an aquatic sample. And that ratio seems to be true across different time points with slight variation by basin. So I think we're largely on trend with freshwater aquatic systems, and we'll look to sort of discern some of these patterns between basins in a bit more detail once our nutrient analyses come in. If we look a little bit now at the taxonomy and functional data from these 3 sites, these are hot off the press. So I'll walk you through them, orient you to them a bit, and then sort of wrap up with some ideas of where we're heading with these as well. So what is grouped here now are just by date for different sampling sites. So these different time points, these different groupings now indicate time points here. And at the bottom, I've indicated they go sort of left to right in trios of Missisquoi Bay, St. Albans Bay, Mallett's Bay. The one unique sample here that's now in that pink color is that the sampling time point in March, there was ice on Missisquoi, but both St. Albans and Mallett's Bay were unsafe to sample that day, unfortunately. So this is sort of the standalone data point. But this is the only sample we captured that was actually iced over in this last winter season. And intriguingly, it does have some pretty substantial taxonomic differences from these other time points, but it is tricky. There's not a lot of weight we can give to those differences since it's this sort of singular time point. It would have been great if St. Albans and Mallett's Bay had also had ice to compare. There do seem to be between basin and also temporal taxonomic and slightly functional differences. However, we do sort of see that general trend we saw from the summer data where even though there are most differences in taxonomic data, there are also some functional data that seem to change a little bit, but less so than the taxonomy. So again, we're going to dive a bit deeper into some of these functional groupings to look at what we can try to correlate in terms of trends of taxonomy and function, trying to build this idea of the microbial ecosystem that's happening in these communities at these time points. And then the next steps with these data are, not advancing, here we go. Excuse me. Using both long-term data sets as well as the physical chemical parameters we collected to start trying to look at some of these associations between taxa and functional groupings. So my students this summer pulled out the long-term data from the DEC. Some of the parameters we're interested in comparing to across our 3 basins. And we can add our nutrient data sort of into this landscape from our time points to start looking at how we might infer longer periods of community assemblies. And another student actually worked on, excuse me, trying to constrain our understanding of ice in Lake Champlain, which is a tricky question. So the NOAA data, which sort of look at did the ice freeze or not across the full lake, are a pretty coarse metric of what's happening for ice on the lake. And so Pete Strafert was using some GIS techniques to try to pull Landsat imagery from our basins of interest to distinguish ice or open water across time. And it turns out this is a really challenging task because many images in the winter are just full of clouds, and clouds look very much like ice in this software system. So right now, when we have relatively low cloud cover in the imagery, we're only predicting ice cover accurately at about 89% of the time. So this is maybe a slight improvement on did the lake freeze or not, but it's not as accurate as we want it to be. So we're still trying to tweak some of these parameters. And this leads to sort of the upcoming work and ongoing work in different ways of thinking about what we know about ice in the system. So I'll just end with sort of the ongoing pieces of these projects. So for the microbial ecology fieldwork and lab work, we're now trying to really get a sense of how do interannual seasonal variability and those sort of parameters impact the microbial ecology, both in terms of the taxonomy and the function. And we're going to also add a metric of productivity to actually measure how productive these systems are over different periods of time. And then I'm really excited, the Sea Grant project that will just be starting this spring is trying to also bring in traditional and community knowledge on what have people noticed over time in terms of winter phenology changes, as well as sort of changes in phenology of blooms. So more severe, more extended, and trying to really bring in local knowledge on, these basins that people have, paid really a lot of attention to over the years. So the sort of big picture drawaway is that my hope is that understanding this microbial ecology can identify sort of nutrient cycles, functional groups, ideas that we might develop potential management strategies or targets for CHAB management in Lake Champlain, as well as with this news piece, really trying to engage more in community work to facilitate learning and ultimately, hopefully, stewardship of this really incredible ecosystem. And with that, I just have a few many folks to thank. So some photo representation of this really incredible, fabulous crew of students who worked on these projects. I want to highlight particularly for the work I shared today, Irene Hu and Eli Dabari, as well as Pete Streifert, who did the ice modeling. And Isabel Sang is here, knee deep in the water, for a lot of the work I presented today, as well as collaborating with Mindy Morales at UVM on this idea around sort of phycosphere microbial systems, and to Jason Stockwell and Marie Pierre, a postdoc with Jason, who've really been helpful in thinking through safely working in winter lake systems. Funding from the Water Center, upcoming from Lake Champlain and Microbial Ecology. And with that, I'd be happy to take any questions.